Topic for today is difference in manufacturing process for tablets and biologicals. So before we understand the difference in manufacturing, first let us try to understand the process of manufacturing of tablets and process of manufacturing of biologicals individually. Then we will combiningly study what are the differences between tablets and biologicals. So when we speak about tablet manufacturing, basically we are discussing about the three processes which are included in tablet manufacturing: direct compression, dye granulation and wet granulation. And you can see these are the steps involved in it: sieving, mixing and then it will go for compression. This is direct compression, wherein we are mixing all the excipients and drug APIs and then we are going directly for compression, hence the name direct compression. Sieving, wherein we sieve all the excipients and APIs, then we mix drug, diluent, glidant and disintegrant. We mix them in the desired proportion for desired time. Then we add lubricant to it so that the excipients or API doesn't stick to the dyes and punches. And then we go directly for compression, wherein we compress the tablets to desired shape and size using dyes and punches. Then depending on whether coating is required, we go for coating and then of course the last stage, packaging. So direct compression is comparatively a very easy method or simple method. Then next is dry granulation, wherein we create small granules of API and excipient. This will depend upon the adherence qualities of API and excipient. Here the excipients and API, they adhere to each other, they stick to each other, hence direct compression can be implemented. When it comes to dry granulation, sieving, mixing, compression, size reduction, screening and mixing. So here we are mixing drug, diluent and lubricant over here. Then we will do compression, not compression into tablets, but still the powder will be compressed. Then size reduction, we need to break it again, then screen it through desired mesh and then mixing the disintegrant, diluent and lubricant, and then we will compress it to desired shape, size, coating will be given and packaging. When we speak about wet granulation, we will need to add binder solution to it. So sieving, mixing of drug, diluent, glidant and disintegrant, then we will do the granulation step, wherein we are adding binder solution to it, because the excipients and drugs will not combine with each other or adhere to each other. So we will need to add binder solution. Then drying, screening, the granulation, since we are adding binder solution, we need to dry it. Screening, mixing, here we are mixing disintegrant, glidant and lubricant, and then we will go for compression, coating and packing. So this was a brief idea about how the tablets are manufacturing. So these are the three processes and the sub processes included in it. So now let us see what are the steps in process steps and parameters involved into tablet manufacturing. So these are the steps, and let us see what are the parameters which we are going to check and what are the controls those that will control our in process parameters. So when it comes to mixing, it is blend uniformity which we need to check, and what will determine it is mixing time and speed. If we mix it for long hours, it will not give the desired result. Speed is also important to have a proper mixing and blend uniformity. Granulation, true density, granule size distribution, moisture concentration, flow rate and mass of the batch. These are the parameters which we need to check during our in processes after the granulation process is done. And what will determine it, or what are the parameters which we need to control, are mixing time, chopper amperage. These are the parameters for granulation. When it comes to drying, we need to have a check on moisture content, and inlet and outlet temperatures are something which we need to control to have the desired quality during drying. Screening, particle size distribution, and what will determine the accurate size of screen. So screen size is important for it. Then is the lubrication. When it comes to lubrication, now we are moving the stages towards compression and final stage. So now from here onwards we are more concerned about assay, or during next stages we are also going to see about related substance. So blend uniformity and assay are the parameters which we are going to check after lubrication is done, and the parameters or controls what determine it is sieve size, blending type, and rotation per minute. When it comes to compression, content uniformity, weight variation, hardness and thickness are something which we need to check, and the compression force is a process parameter for it. Coating, assay, dissolution, related substance and friability. Spray rate, inlet and outlet temperature, pan speed. Spray rate because we don't want it to have more coating at the same time, lesser coating, and inlet and outlet temperatures. When it comes to temperatures, the excipients will have effect of temperature. Pan speed, more the speed it will not coat properly. Even when it is lesser speed, it will not coat uniformly. So these are the controls which we are going to check, and the test what we will be doing after our coating is assay, dissolution, related substance and friability. So this was about tablets. It's in process step. So we have completed understanding the manufacturing of tablet and its in process tablet. Now we will move to biologicals. Now let us try to understand about biologicals. How are the biologicals manufactured? So let us first start to understand how is the API manufactured, or how is the starting process of it. First is source DNA. This is a source DNA. It can be a human DNA or biological DNA, or it can be artificially manufactured target DNA. So this is a source DNA in which we have a target DNA. This will depend upon what type of therapy we are searching for. For example, if we are trying to tackle with diabetes, obviously it will be insulin. If it is osteoporosis, it will be parathyroid hormone. So in that, it will be the sequence of amino acid. For example, when it is parathyroid hormone, it is 1 to 34 amino acid sequence when we are dealing with osteoporosis. So this is our DNA of interest. It will be clubbed with a vector. This is known as a vector. Vector is nothing else but it is a vehicle which will be then added to a host cell. This is a host cell. So first this target DNA will be included in a vector. This is known as a vector. Cloning into DNA vector. So this is a vector and this is a target DNA which is detached from here and it is added to vector. Vector is nothing but it is an extra chromosomal material which is having capability of multiplying. Because this target DNA cannot multiply of its own, it needs to be added to vector, and this whole is known as expression construct. This will now go into host. Host is generally a E. coli or any other microbe which has a capability to multiply the expression construct. So with this they will multiply and then we will get a number of this. This is something which will multiply. So this whole process is known as cloning. So we have one target DNA, out of that we have created number of copies of it because this is something which is going to be incorporated in our finished formulation. We have now seen how the cloning is done. Now let us try to understand how is the actual manufacturing and what are the steps, what are the purification steps which are performed during manufacturing of biologicals. So as we understand, we have DNAs and we have introduced the vector in the host cell, and now we have many target DNAs. So obviously it is going to be in a petri dish. From that we are going to make finished formulation out of it. So this is the array of steps what are involved. From cell bank, then we will grow it with the help of medium, with the help of other ingredients. Then we will get so many that we now have it in flask. From flask it will be in seed bioreactor. From seed bioreactor it will be in production bioreactor. So here then we are controlling all the nitrogen, oxygen supply and other vitamins, and then we are centrifuging it. Now from this we can understand that small quantity of target DNA are now growing into a much larger scale, and then we need to do centrifugation wherein we will get very small part of it. From centrifugation is separation of target DNA from other impurities. Then to further filter it more, we will perform depth filtration wherein we will filter the solute and we will get more concentrated DNA. So now we need to harvest it. Harvest is only the interested DNA or our target DNA, which is the DNA of interest. We need to separate it from rest of the materials. So that method is known as harvesting. After harvesting, so here we have increased it. Now no more of increasing DNA in number, but now whatever DNA we have received, we need to separate it from rest of the impurities. So this is a step in the center, and then now all the steps what we are seeing over here are related to purification to get the more specific target DNA which will have very less number of impurities. So here comes the affinity chromatography, wherein we will have it on the basis of the affinity, the particle size, we will perform this chromatography. Then viral inactivation, because as we discussed, these are biological sources. We don't want any unwanted virus to get into our finished formulation. So viral inactivation is done with the help of some solvents or detergents or acid wherein low pH. So here the viruses will be inactivated. Then comes ion exchange chromatography, wherein in chromatography we will separate the DNAs and impurities based on the charge of it. So ion exchange chromatography. Then viral filtration. Whatever virus are there, we will try to filtrate it to get more pure API. Then diafiltration or ultrafiltration, wherein with the force the solute will be passed through membranes. Then bulk fill, and from bulk fill it is finished formulation. So here we are getting our API, and then from here we will add preservatives to make it more stable, or stabilizers will be added, and then we will get it as a finished formulation. So this was a brief regarding the manufacturing of biologicals. So now let us try to understand what are the in-process involved in biologicals manufacturing. Steps, in-process parameters and process control. As we have understood for tablets, now let us try to understand for biologicals. So these are the steps what we have studied, and then now these are the in-process parameters. For cell bank, vial and flask, it is viable cell density, cell viability, cell morphology, bioburden. So whatever collection we have got, we need to understand it whether it is of desired cells. So we need to understand cell morphology, whether the cells are viable or they are inactive, non-viable, bioburden. So we need to understand if they carry any adventitious agents or not. Process controls are definitely pH and temperature, which we are going to control. So if you see here carefully, most of the places pH and temperature plays a very crucial role in having the in-process parameters in the desired quality. Then when it comes to bioreactor, we need to check the viability, cell density, viable cell density, cell viability, cell morphology, bioburden and adventitious virus. And of course the process control will be pH and temperature, and since this is bioreactor, we also need to have control on dissolved oxygen quantity. Harvest, Mycoplasma adventitious virus. Mass and flow will be the controls which we are controlling. During affinity chromatography, bioburden, mycoplasma and bacterial endotoxin. Since these will be injections, we need to have bacterial endotoxin as a parameter, which is highly important. And the controls what we will be doing is elution buffer and pH. So because in affinity chromatography, as the solutes elute, we need to understand the quantity, the elution buffer which we are using. Viral inactivation. For viral inactivation, of course, bioburden and bacterial endotoxin will be the parameters, and low pH, because for viral inactivation it is a low pH or solvent which will be using. Time and temperature. These are the controls which will control the bacterial endotoxin and bioburden. Ion exchange chromatography. During ion exchange chromatography, we need to check what is the main peak percentage, bioburden, bacterial endotoxin. Column load factor, buffer preparation, mean peak and collection criteria will be the controls which we will be controlling. Then for viral filtration, filter integrity test, because this is a filtration, so definitely filter integrity test needs to be checked. And for that, pressure load, inlet pressure load and pH will be the control factors. Ultrafiltration and diafiltration. So over here it will be protein concentration. This is a protein, nothing but the protein of our interest. Protein concentration, bioburden and bacterial endotoxin. pH, osmolarity and diafiltration volume, because volume also determines the quality of the filtration. Then is the last step, which is formulation. Here protein concentration, identity, purity and bacterial endotoxin. These are the tests which are very much essential when it comes to injections. pH, buffer formulation. These are the parameters which are going to, these are the controls which will be requiring for checking parameters. So we have seen how the manufacturing of tablets is done. We have seen the in-process steps in tablets, what are the biological manufacturing steps and in-process during biological manufacturing. Now coming to the last topic about understanding the differences or jotting down the differences between tablets and biologicals. And the parameters to understand these differences are starting material or API and manufacturing equipment, in-process step, analytical techniques and regulatory requirements. So let us try to understand what are these. In tablets, of course, chemicals will be used and biologicals are biological sources, DNAs or host cells, all are biological materials. When it comes to manufacturing equipment, we will use blenders, granulators of different types, rotating the granulators and dryers, fluid bed dryers and different type of dryers will be used, rotary dryers, compression machine can be many stations or single station. Then when it comes to biological, we are using small vials or petri dish, and then we are gradually increasing the size of our interested DNA material. So vial, petri dish, bioreactor, fermenter and centrifuge machine. These will be the equipments what we use for biological. When it comes to in-process steps, here are all the chemical analysis steps which we will be using. Particle size distribution, assay, content uniformity, disintegration. But here our focus is on viral inactivation, affinity chromatography and other different types of chromatographies. Analytical techniques what we generally use for tablets will be HPLC, GC, atomic absorption spectroscopy. But when it comes to biological, it will be ELISA, SDS, PAGE. When it comes to regulatory requirements, it will be BE or comparative DISO, which is of prime importance. But here we will need to do head-to-head comparison. Head-to-head comparison is comparison of toxicity and preclinical or clinical trials. So that we need to do. Also analytical comparison we need to do between the reference and our test biological. Then stability, bacterial endotoxin and sterility. These are the important regulatory requirements, because these are basically injections. So these are critical here. Sterility, bacterial endotoxin and viral inactivations are of prime importance. So this was basically the difference between the manufacturing process of tablets and biologicals. Thank you.